that. This Embryology Highlights screencast, Head Development 3, presents the development of the mouth and its contents. The mouth originates as a stomadeum. The mouth originates as an embryonic stomadeum, a cavity produced by growth of jaws from the first pharyngeal arch. Also, the frontonasal prominence contributes the upper incisor region to the stomadeum. The stomadeum is lined by ectoderm. It is separated from the pharynx by an oropharyngeal membrane that must degenerate for the mouth to communicate with the pharynx. During development, the oral cavity communicates with the nasal cavity until palates grow to separate them. This image presents an anterior view of the developing stomadeum. An early stage is shown at the left and a later stage is shown on the right. The frontonasal prominence, here colored blue, will form the nose and nasal cavity, including the incisor region of the stomadeum. Maxillary and mandibular processes from the first pharyngeal arch form the upper jaw and the entire lower jaw, including the mandibular symphysis. Initially, an oropharyngeal membrane separates the stomadeum from the pharynx. That membrane must degenerate in order for the mouth to communicate with the pharynx. This image shows on the left a mid-sagittal view of a developing embryo. We can see the oropharyngeal membrane that separates the pharynx from the face. Here we see a surface view of an embryo. The blue frontonasal prominence will form the nose and incisor region of the stomadeum. The first pharyngeal arch will grow out and form the upper and lower jaws. On the right, we see 3 stages of development looking at the embryo from an anterior view. Here we see the frontal nasal prominence, which is growing out and contributing the incisor region of the stomadeum. Meanwhile, the first pharyngeal arch develops a maxillary and mandibular process to form the jaws. The mandibular process will meet at the midline and create the mandible and the mandibular symphysis. The maxillary processes will only grow as far as the canine teeth, and the medial nasal processes will fill in and provide an incisor region. So growth of jaws and the nasal processes forms the stomadeum. The ectodermal lining of the stomadeal cavity develops a labial gingival lamina, which invades the underlying mesenchyme, becoming a labial gingival groove. That groove then becomes the adult oral vestibule, which separates lips from gingivae. Caudally, the lips fuse to form cheeks. This cartoon illustrates that process. Here we're looking at the floor of the stomadeum, and we see that the ectoderm has formed 2 lines of thickening, which are called laminae, and we're looking at the outer lamina, which is called the labiogingival lamina. If we take a transverse section and look at that, we see this. Here's the surface ectoderm, and there are 2 thickenings, so we're concerned about this outer thickening, the labiogingival lamina. We see that the thickening invades into the underlying mesenchyme, and then it undergoes a cavitation. So we end up with a separation between the adult lip and the adult gingiva, and that is the oral vestibule. Teeth are formed from that internal thickening of ectoderm called the dental lamina. Periodically along that, there is a proliferation of cells forming dental buds. So here we see this internal lamina in transverse section, and periodically we have this thickening that forms a dental bud. Each dental bud gives rise to a tooth in the following way. Each bud develops into a cup-shaped enamel organ. The enamel organ induces the underlying ectomesenchyme to become a dental papilla. The enamel organ also induces the the epithelium on the surface of the dental papilla to become odontoblasts that are capable of depositing the dentin of the tooth. Meanwhile, the odontoblasts of the dental papilla induce the surface of the cup to form amelioblasts, which are able to secrete enamel. And so as you look down here, we have an amelioblast layer opposed to an odontoblast layer, each secreting the enamel and then the dentin, respectively. And so the two layers are separating and producing a tooth in between. It is significant to note that if this is a deciduous tooth, the permanent tooth also forms at the same time in the embryo, but the permanent tooth remains dormant until a certain time when it starts to grow and erodes the root of the deciduous tooth and eventually pushes it out so the permanent tooth can erupt. But the point is that any damage early in development and postnatally will affect the permanent tooth as well as the deciduous tooth because they're both located at the same place. Meanwhile, the ectomesenchyme that is surrounding this tooth formation will form a dental sac, and that gives rise to three layers. First of all, bone that encases and becomes the alveolus and encases the embryonic tooth, and a periodontal ligament which anchors the tooth within the alveolus, and also cementum, a material that coats the root of the tooth as opposed to the crown of the tooth, which is coated by enamel. So this illustrates what we just said in color. We have a dental lamina, which forms an arc of ectoderm thickening on the surface of the stomadeum. There are periodic thickenings that form dental buds along that arc. The dental buds induce the underlying ectomesenchyme to form a dental papilla. Meanwhile, the dental bud forms an enamel organ, which is cup-shaped. The enamel organ induces the dental papilla to form odontoblasts, here shown in yellow. So this is the dental papilla, and these are odontoblasts, which are capable of secreting dentin, which is shown and striped here. Meanwhile, the dental papilla induces the enamel organ to form amelioblasts, this red layer, and the amelioblasts are capable of producing enamel, and so the two epithelial layers are producing their products, which creates a dentin enamel-coated crown of a tooth between the two layers. Tooth enlarges as the two layers separate. Here we see the permanent tooth in a dormant stage, but located right next to the deciduous tooth. Here we see an actual image of a developing tooth. This is the surface epithelium. We're looking at a section through the gingiva. Here we see an enamel cup which is lined by amelioblasts on the surface, and it has induced the ectomesenchyme to form a dental papilla, which is lined on the surface by odontoblasts. So these two epithelial layers will secrete their products, and a tooth will develop between them. Here's an image of a tooth further along. This would be the dental papilla region here. It's lined by surface epithelium, which are called odontoblasts because they're capable of producing dentin. This red tissue here is the dentin. It lines up against enamel prisms, which are being formed by a layer of amelioblasts, which are lining the concavity of the enamel organ. The tongue develops from swellings on the floor of the pharynx. The body and apex of the tongue comes from paired distal swellings. The root of the tongue comes from a proximal swelling. Also, pharyngeal arches contribute to the tongue development. The first pharyngeal arch contributes general sensation, and the trigeminal nerve will supply a branch to the tongue. The second pharyngeal arch contributes a taste sensation via the facial nerve, and the third pharyngeal arch will supply the root of the tongue via the glossopharyngeal nerve. Muscles of the tongue originate from occipital somites. Those myotomes migrate to form tongue muscles, and they are innervated by the hypoglossal nerve. This cartoon shows the apex and body of the tongue in green because the bilateral distal swellings form it. They receive contributions from the first pharyngeal arch. Meanwhile, the root of the tongue forms from the proximal swelling on the floor of the pharynx, and it has contributions from the third pharyngeal arch. Here we see a cartoon of human tongue development. We have the distal swellings and the proximal swelling. It shows that from the pharynx, that the apex and body of the tongue grow into the stomadeum, and thus they pick up an ectodermal coat. So the apex and body of the tongue are coated by ectoderm, and the root of the tongue remains endoderm. This image shows the distal tongue swellings and the proximal swelling. There's also a middle tongue swelling, which really contributes to the ungulate tongue. It doesn't contribute much to the carnivore tongue. Here we are contrasting a goat tongue with a cat tongue. And you see in the goat, the ungulate, we have this prominent torus linguae that is produced by that middle tongue swelling, not evident in the cat tongue. Salivary glands are derived from ectodermal proliferations in regard to the parotid and zygomatic gland, and the endoderm proliferations produce the lower salivary glands, mandibular and sublingual. Salivary gland development is typical of embryonic gland development. Gland development begins from epithelial proliferation, which grows and invades into the underlying ectomesenchyme in this case. These are solid cords of cells then that are going to grow and invade. They will branch, and at the ends of the branches there will be enlargements which form the future acini. So the entire gland grows in a solid fashion, which enables it to penetrate into the underlying mesenchyme, and then eventually the gland will have to become hollow. The acini form the secretory portions, and the elongated branches form the ducts. The main duct will open on the surface where the initial growth took place. However, if that surface moves, shifts during development, then this duct will elongate, still opening at that site, but it may be at a distance from the original gland. Glands that are polystomatic are actually multiple glands that are all clustered together and regarded as one gland in gross anatomy. This illustrates a more colorful image of what we just saw. The surface epithelium, either ectoderm or endoderm, will proliferate, will then continue to grow as a solid core of cells. It will then branch into the underlying mesenchyme or ectomesenchyme. Continued growth and branching will ultimately lead to acini or swellings produced at the ends of each branch. This whole process then will have to hollow out, undergo a cavitation to produce a functional gland. The acini then producing glandular material, and the duct system formed by the branching. If this hollowing out process is incomplete, so there is a stenosis or an atresia at a certain point, then we would expect a cyst to form, because the glandular secretion is working fine, but the secretory products cannot escape to the surface. This image illustrates the zygomatic and parotid salivary glands, which are ectodermal in their origin, and the mandibular and sublingual glands, which are endodermal in origin. The zygomatic appears to be a polystomatic gland with multiple ducts opening onto the surface. The others are monostomatic, and you can see that the original opening has migrated with growth of the jaws, so that the duct has had to elongate. So this site was originally in the vicinity of the gland. The adenohypophysis is one part of the hypophysis, which is the pituitary gland. There is a neurohypophysis, which comes from the brain, and the adenohypophysis will come from the stomadeum. It will begin as a placode and then bud off and become a pouch, detach from the stomadeum and wrap around the neurohypophysis. The hypothalamus, which controls the hypophysis, can do so directly in regard to the neurohypophysis, but the adenohypophysis has no connection with the hypothalamus, so the hypothalamus will have to control the adenohypophysis via the bloodstream. So here's a color image of hypophysis development. We have the pharynx separated from the growing stomadeum by an oral neuropharyngeal membrane, which we'll have to break down. But here in the dorsal wall of the stomadeum is a placode beginning to form. Here at a later stage, we see the neurohypophysis growing as an outgrowth of the diencephalon, and we see that placode has become a pouch which has detached from the surface stomadeum and is approaching the neurohypophysis. And here we see the adult stage. The adenohypophysis from the stomadeum is wrapped around the neurohypophysis. The hypothalamus can control the neurohypophysis directly via axons, but it has no connection with the adenohypophysis except through blood vessels. So this concludes this embryology highlight screencast, Head Development 3, dealing with the stomadeum and its contents.